Good day, this is Professor Will, CBMD PhD, and today's Wednesday, May 4th, 2022. It is 5:17 and 17 seconds, a.m. Eastern Standard Time. Today's research, oral cancer. So let's hop to it here. Could blocking or deleting a protein help prevent common oral cancer? Date April 25th, 2022. Source, Boston University. Around 1/3 of people with oral squamous cell carcinoma don't survive it. Potential researchers have found deleting or inhibiting a protein in the tongue might stall tumor growth. The most common head and neck cancer, oral squamous cell carcinoma, often starts off, as many other cancers do, quite innocently. Perhaps as a little white patch in the mouth or a small red bump on the gums. Easy to ignore, to downplay, but then something changes, and the little blotch becomes more obvious, starts growing, burrowing into connective tissue. Patients who are lucky enough to see a dentist before things take a nasty turn have a shot at being able to prevent the lesions from turning cancerous, or can at least make sure treatment starts when it is most effective. But for those who aren't that lucky, the outlook can be bleak. The 5-year survival rate of oral squamous cell carcinoma is around 66%. More than 10,000 Americans die of oral cancer every year. Smokers and drinkers are hardest hit. The researchers have found that dialing back, or even genetically deleting, a protein that seems to spur the cancer's growth might help limit a tumor's development and spread. They say their findings make the protein, an enzyme called lysine-specific demethylase 1, a potential druggable target, and something that doctors could aim chemo and immuno-oncology therapies at to downtake a tumor. Given that at least 1/3 of Americans don't visit a dentist regularly, according to the Centers for Disease Control and Prevention, the discovery could be a future lifesaver for those who miss out on preventive care. And these findings have significant implications for new and potentially more effective therapies for oral cancer patients. And this study is an important step toward the development of novel groundbreaking therapies to treat oral cancer. Research began testing what would happen to tumors in the tongue if lysine demethylase 1 was blocked to restrict the enzyme. The researchers either knocked it out by manipulating genes so lysine demethylase 1 is effectively switched off, or used a type of drug called a small molecule inhibitor, which enters the cell and impedes its normal function. Already in clinical trials for treating other cancers, small molecule inhibitors haven't previously been tested against oral cancer. Bias found that disrupting lysine demethylase 1 curbed the tumor's growth. The aggressiveness or bad behavior of the tumor went down. Researchers found that when they inhibit this protein, it promotes anti-tumor immunity. Our body tries to fight by itself. But lysine demethylase 1 isn't the only troublemaker in the tumor. When it's upregulated, it messes with the cell communication process, the hipposignaling pathway that normally helps control organ growth and tissue regeneration. Research says hipposignaling pathway, lysine demethylase 1, and a couple other proteins then get stuck in a vicious cycle, each one pushing the other into increasingly aggressive and harmful moves. We need to break this cycle. And to find a new way of doing that, the researchers coupled the effort to inhibit lysine demethylase 1 by targeting hipposignaling pathway with a different inhibitor, a drug called drwillphd, originally developed to help treat serious eye conditions like macular degeneration. Drwillphd is being tested by other researchers as a potential cancer treatment, including ovarian cancer. The combination proved effective According to researchers, they also threw a third drug into the mix. Researchers say using lysine dimethylase 1 inhibitor in combination with a common immunotherapy drug that helps white blood cells in the immune system kill cancer cells. An immune checkpoint inhibitor called anti-programmed Death1 ligand antibody showed a favorable response. The findings provide a basis for future clinical studies based on the inhibition of lysine dimethylase 1, either as monotherapy or in combination with other agents to treat oral cancer in humans. Although the studies are preclinical, restricted to mice and some human tissue, researchers want to expand the look at human clinical trial samples. The researchers focus on the biology of oral cancer may also help make the development of other future treatments more efficient. The people get very excited when you have a drug that may show some positive preliminary results, but very frequently these studies move forward to humans, cost billions of dollars, and then eventually fail. And if you really understand what pathways, what cell processes are impacted by these inhibitors, then it allows you to predict in advance whether something is going to be successful in human patients. And so we can think about disease interception and perhaps think about preventing the tumor from happening. And with access to a clinic, as well as a head and neck surgeons from neighboring hospitals, researchers can test any new treatments and approaches on human tissue samples. Researchers say of the human samples, they can interrogate them for responses to small molecule inhibitors by capturing tumor slices and trying to treat them with different inhibitors to see the response. Eventually, it can also open the door to personalized precision medicine with researchers trialing different therapies on tissue from individual patients. And then it will predict whether this person can be treated with this study. And this is something they really want to develop. Research will be able to say, this is precancerous or cancerous. It will impact their diagnosis. And then, in terms of therapy, because they are now aware of what can work, what immunotherapy can work, they can make specific reference to where patients should go next. It can improve the quality of diagnosis and treatment in the long term. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.